right, welcome back. We have a guest today. As you can see, I'm presenting because Dr. V evidently is too lazy to do so. I'm going to do the presentation, and I'm not from here. I'm from down the street a little bit farther. Anyhow, we just get started here. Animal cells. We're going to look at various aspects of the animal cell, at least the human ones. We're going to look at plasma membranes to begin with. Plasma membranes, of course, are made up of two layers that fill a variety of pores, molecules, and channels. It's sort of the interface, if you think of it as the analogy, are the doors and windows and things, and then people's houses and or aircraft, UFO doors and that sort of thing. Holds the contents of a cell in place, takes food in and nutrients and various other things, aids in building molecules that are embedded that can transport things in and out. You can see all this elaborate stuff up here. And plasma membranes are the gatekeepers. And so they essentially are the doors and windows and various things to allow you to get in and out of the building. And it can be very selective, so keep that in mind. All made up of a very simple concept that we've already talked about. It's the phospholipid bilayer. We have the polar head, which is attracted to water, and it's made up of the glycerol, and linked to that are these hydrophobic tails, those long tails that make up sort of the spontaneously form these bilayers. But in membranes, you can see that the like dissolves like, so the hydrophobic, water-fearing, tend to align with other water-fearing type and will form this phospholipid bilayer spontaneously. So that's the deal. Phospholipids appear to have a head with two long tails. So the glycerol molecules is linked, containing phosphorus, and this makes this region more polar, so it tends to like water. It's attracted to water. And then the long chains are the nonpolar, which are just the carbon and hydrogens we've talked about. Phospholipids are not locked into place. That's really key if you remember looking at the video, the movie. What this does is allows it to float, sort of like buoys in the ocean, and we get this constant floating that's going up and down, and because of that, it's makes it quite interesting. Chemical structure of phospholipids, as we've already talked about, as a key point, we have the hydrophilic and hydrophobic regions. Hydrophilic love water, phobic hates the water, fears the water, and they're arranged tail facing each other in a phospholipid bilayer. And that's a very interesting concept because it's used throughout the cell. Transported materials are kept in a bag made up of this phospholipid bilayer, so it immediately melds and heals once it's exposed to another bilayer, and so they meld perfectly. It's the perfect 100% reusable way to do your shopping. Every cell, living organism, is enclosed by this membrane, so it's universal. All living things do this. Molecules are embedded in this. It's really important. So we have ways to communicate, ways to transport things. We can add glycerol, which will stiffen up the membrane if we want to. We can actually build rafts, so various different or similar things can float together and form that raft. And I think it's really, really interesting. The function of plasma membrane, again, if I delineate the aspects of the things floating in it, we have the carbohydrate chains, the cholesterol for stiffening, transport proteins, membrane proteins for various reactions and sites at things that need to get done, The transport moves things in and out, as I just mentioned. Extracellular fluid, we can bind these sorts of things and regulates processes within the cell, sort of as a communication. Then we have recognition proteins, a fingerprint for identification by other cells. And we'll talk about various aspects of these things, but this is really, really important. The membranes, as we've looked at in the class and in the movie, if you look, there's the raft I was talking about. It's cholesterol and related types of proteins, and all of these things float around in this mosaic of this phospholipid bilayer. So that kind of gives you a feel for what I meant by that fluidity of that phospholipid bilayer. Now, there are times when we have faulty membranes, and these can cause diseases, and cystic fibrosis is one of those. It gets a thick and sticky mucus, and it's all due to the fact that one of the pumps, sodium pumps, aren't working properly, and the mucus thickens. And it's an inherited disease. It's just as simple as a transport mechanism can cause all of these really serious issues. And so I just wanted to point that out, is that sometimes we take for granted some of these basic things, and we find that we have problems. So, summary, 4 primary different types of membrane proteins, each of which performs a different function. Receptor proteins bind to chemicals in the environment. Recognition proteins give each cell a fingerprint. Short branch carbohydrates are attached to proteins and heads outside of the membranes that serve as part of the fingerprint along with recognition proteins. So all of these things play into recognition, and it's an important part of that fluid mosaic. So the plasma membrane can also contain cholesterol, which helps the membrane maintain its flexibility. It stiffens it. It prevents the membrane from becoming too fluid or too floppy, and it also adds this antifreeze-like characteristic. So we've seen overall the plasma membrane is a mosaic. It has the proteins embedded in them that do all sorts of different functions, which we'll talk more about at a later time. So for this section, I think we survived it, and perhaps you'll have your standard lecture back. Until next time.